Let's welcome our next speaker. It's Yihang Jiao from Monash, if I'm not mistaken. No, it's the University of Melbourne, sorry.
Hello, everyone. I'm Yihang Jiao, and today my topic is to introduce the resistor-capacitor circuit models for predicting the transcranial magnetic stimulation propagation in the human connectomes. I have no disclosures, and here is our outline today. I will start with the motivation. So our motivation is to predict the TMS propagations, and we know that the TMS is a non-invasive technique which can precisely deliver the current into the brain. And the DLPFC is a commonly used target for the depressions. We also know that the TMS will not only activate the local targeting region, but the signal will propagate through the whole brain networks. So if we can better predict such propagation, we can provide some more effective and personalized TMS treatment. There are some existing models like the neural math or some graph communication models for predicting such propagation. However, those models, well, you can see that there is a trade-off. Like for the neural math, they can capture a really high tempo dynamic resolution, but lack for the spatial resolutions. For the graph theory, it can capture some really high spatial resolution, but it just lacks the ability to capture the dynamic one. So we wish to establish models which can combine both high spatial and also temporal resolution. We borrowed the idea from the very basic electrical circuit. We know that the gray matter voxel will contain a lot of neuron cells which have the ability to store some charge. And this is equal to the capacitors in a circuit. So you can imagine that if you have more neurons or the larger gray matter voxel size, volume, you will have the much more ability to store charge, which will let you have a larger capacitor values in the circuit. As for the connections or the white matter tracks, you know that it has the ability to transform the signal from node to node. And it's very similar as the resistance inside a circuit. And how do we calculate such resistance values? We can use from its cross-section and the distance. So luckily, we have our structural connectance weight, or the count numbers, and also the distance track of from the white matter track. In that case, we have our structure network, and then we take every node into a capacitor, and between the connections, we just turn them into the resistance. And we turn our brain network into some really simple resistant capacitor circuit. From this circuit, if we know the initial state of each node's voltage, we can then use some differential equations to solve, during time changes, how is the voltage or the current changes through the whole network. So it's a really simple model. So we start with identify the injection or the injection E field or the current field delivered by the TMS. And the second step is from the diffusion MRI, we can get the structural connectome weight and also the distance. And from using that matrix, we can get our value of the resistance values and the capacitor value and build our circuit models, get the dynamic change map of the TMS signal. And the last step, we can also comparison with the empirical data. So I will show you our model's result. So we apply a single pulse TMS at the DLPFC for setup. Our structural connectome matrix are from the HCP datasets and also in a group level. So this is the dynamic current changing map from time 0 to about 400 milliseconds. So we can see that the current will start flowing from the DLPFC into the direct connections regions at the very earliest stage, and then it will separate to some like long distance or indirect connections with DLPFCC regions at a later stage. And like we can also plot like the current curve for each node separately. You can see that there are some different types of nodes. So for example, the right DLPFC, you can see that it will receive a strong current at the beginning time, but for some later, like other nodes, like the SGC, the motor cortex, or some visual cortex, they will receive no current at the beginning, but in the later time, it will receive some different amount of current depending on the connectome between them. And if we take the whole, the full simulation period's maximum current, we can get some steady state map. In this case, we take the maximum current map, and then we can plot such like steady state map, which we can use for our validation with the real world, like concurrent TMS FRMI datasets. So in this case, we compare with two different groups, concurrent TMS FRMI both signal map. For the first group, we can see that we directly compare with our model with the FRMI, and we can see a significant and positive, like R equal to 0.41, a very high like correlance between those two, our model and the real empirical data. But as we can see for the second map we use from TIG groups, there seems to be not a significant correlance. So what caused that? We looked into their data and we found that there is a large TMS auditory artifact inside. So what we are thinking is like, if the TMS auditory artifact is like, because you know that the TMS course will generate some loud kick sound when you stimulate the target. So it will also activate the auditory cortex. And this one will also have some downstream effect during the propagation. So instead of like directly remove the auditory cortex only, our model can also handle for like multi-site co-founding like stimulations. So in the below rows, we just co-founding stimulate the auditory cortex and also the DLPFC. And then we ran the simulation and compare with the real empirical data again. Well, the result seems much better. It's significant and with a R value equal to 0.4. So yeah, that's some really positive result. And sorry. Yeah, here is what our models can do, all the strengths. So the first one is that, as we show, we can provide some high resolution, both temporal and also spatial precise on predicting such stimulation propagation map. And as we talked before, our model can handle with, for example, the co-founding like stimulation targeting regions, and also for here, instead of using a single pulse TMS, we do some RTMS. We apply a triple 10 Hz simulation at the DLPFC. So the pattern is totally different with the single pulse. So that's what our models can do. And the third one, and I want to mention, is that because our model is basically depend on the electrical characteristics of the neurons, so it's like biophysical interpretable. So also every model will have its own limitation. Our model also has. So our model is based on some very simple or maybe oversimpled circuit model for the neurons. So it's a linear and passive model. It will cause some strong local effect. So for the future improvement, we can add some more like the complex circuit models, like time-day terms, or adding some gate or some other things, which can make our model more accuracy. And another one I want to mention is that we only chose two data sets to compare to validations. So it's a really small subject amount. And also, we only have the group level structure connectomes. So maybe in the future, we can use some individual level of the structure to build our models. At last, I want to thank my supervisor, Andrew and Robin, and all my collaborators and all the labs. I also have a poster this afternoon, and I'm really happy to see you then, and feel free to contact me. And at last, thank you for listening.
Thank you so much. Nice visualizations. Yeah. We got a question in the app, and this model looks tight. Are you using parcellated data, and does it scale well to vertex and voxel-wise maps?
So actually, our model is basically using some high-resolution HCP structure matrix. So the original resolution of that is based on some voxel space, and it's like 91K nodes involved. But in order to speed it up, for our runtime is for each simulation we just have 10 minutes for runnings. So we downsample the size to 9K nodes. So yeah, if I answer these questions.
Yeah, I hope you answered the questions.
Yeah.
If not, the person, please
yeah.
come and ask him again. There's also, it's the same for the other question in the chat. Please catch him later.
Yes.
And let's thank you again.
Thank you.